Okay, guys. This is our first lab test review over blood. All right, and I wanted to caution you, as always, not to use these lab test reviews as a substitute for studying material in the lab manual and, of course, performing the activities associated with that lab. Remember that you're responsible for the material covered in the lab manual. That is considered fair game in terms of what sort of questions we can pull for the lab quizzes and for the practical exams. Okay, so one of the things we want to discuss here is the idea of the circulatory system in general. Okay, the circulatory system is an answer to a problem in evolution, and that problem is that we started out as single-celled organisms way back when life first appeared on the planet. And back then, it was easy to get nutrients and dispose of wastes because we just used mechanisms like membrane transport, and we lived in an environment that was relatively uniform. And then as the Earth began to change, different environments began to crop up. Parts got dry, parts got wet, parts got salty, parts got less salty, temperatures varied, and so if we didn't adapt to the situation that was occurring on the ground, we weren't going to live long enough to survive. So one of the first things that happened were that these single-celled organisms began to associate with each other in loose groups that are called biofilms. And in those biofilms, you sort of had division of labor. You had organisms that were good at building a home, usually a film that was attached to something like a rock or some other type of surface. You had organisms that were good at defending against attack. You had organisms that were good at finding nutrients and supplying them to the rest of the community. And as a result, this biofilm, and there are still biofilms today, was more effective at surviving than a group of single organisms would be. Okay. Over time, what happened was that these biofilms developed into the first tissues. A tissue is just a group of cells with similar characteristics that can do something that a single cell can't. And the tissue types are connective epithelial, muscle, and nervous tissue. Okay? They all have different jobs to do. Because of that, because of the arrival of tissues, we began to have a more complex body plan, and we had to have a way to deliver oxygen and nutrients to tissues that were far away from the respiratory and the digestive system. And that's where the circulatory system evolved into being, right? That was the purpose, and that still is the purpose that it serves. It's made of the heart, the blood vessels, and the blood that they carry. The heart is the blood pump, right? The vessels are the avenues that the blood takes around the body, and the blood, of course, is the liquid connective tissue that is carried around the body by the beating action of the heart. Functions include transport, again, of oxygen, nutrients, waste products, immune components, hormones, okay? Protection through providing an ability for immune components to get from place to place in the body, and also having immune proteins in the blood plasma itself, and regulation of things like pH and fluid balance in the body. So very important. Also is an important player in thermoregulation. Okay? So all of that we get from the circulatory system. Okay? The cardiovascular system refers only to the heart and the blood vessels, while the circulatory system is the whole thing. The study of blood is called hematology, right? And then we've detailed what the circulatory system does. Adults have 4 to 6 liters of blood, which is a liquid connective tissue that's made up of cells and an extracellular matrix. So the extracellular matrix in blood is called plasma. It's a liquid. Okay? What's in plasma? Well, lots of stuff. There's proteins, there's electrolytes, there's waste products, there's gases. Okay? And then suspended in the plasma are the formed elements, which are the red blood cells, the white blood cells, and the platelets. Okay, we call the red blood cells erythrocytes, the white blood cells leukocytes, and the platelets we call thrombocytes. Okay. Now, among the leukocytes, there are 5 types, and they fall into 2 categories. Right. The granulocytes are made up of the neutrophils, the eosinophils, and the basophils, and the agranulocytes are the lymphocytes and the monocytes. Now you might wonder, okay, what do you mean granulocyte and agranulocyte? What are you referring to here? Well, cyte means cell, okay, and granule refers to a structure that is visible under the microscope that stains with a particular dye, okay? So the granulocytes contain membrane-enclosed vesicles that stain with a particular dye used to stain blood called right stain. And in the right stain are a red dye called eosin and a blue dye called methylene blue. Okay? So the names for the granulocytes actually come from the dye that attaches to the granule. The eosinophils get their name from the fact that the red dye eosin stains their granules more effectively, and the eosin is part of the right stain. The basophils get their name from the fact that the basic dye methylene blue stains their granules more easily, and the neutrophils get their name from the fact that both of the dyes stain their granules equally. Okay, so the basophils have blue granules, the eosinophils have red granules, and the neutrophils have pink granules when stained with right stain. The agranulocytes do not have staining granules, okay, and they include the lymphocytes and the monocytes. Right? Now, what do each of these cells do? Well, neutrophils are bacteria slayers. Right? What they do is they go around, they eat bacteria through phagocytosis, and they also release chemicals in their granules that are antimicrobials. Okay? The eosinophils turn off inflammation, and they also attack parasites, and they also are involved in allergic responses. Okay? Their granules contain antihistamines and antiparasitics. All right? Histamine is a compound that initiates inflammation, so antihistamines turn it off. And then basophils have granules that contain heparin and histamine. The histamine turns on the inflammation, and the heparin prevents coagulation. So what the basophils do is they help to bring blood to the area of the infection, and they also keep the clotting from setting up so quickly that the other white blood cells can't do their work of fighting the infection by providing, a liquid arena in which to engage in battle. The lymphocytes are the agents of cell-mediated and humoral immunity. You might say, well, what is that? Well, cell-mediated immunity is where lymphocytes that are called cytotoxic T lymphocytes show up at infected cells and tissues and blow holes in them and kill them. Okay? So they attack infected cells. And then B lymphocytes make proteins that are called antibodies. And what antibodies do is they latch onto molecules on the surface of a pathogen or cloak a virus or cloak a toxin, and they can also cross-link viruses and toxins and pathogens to each other, and that basically keeps them from being effective at their job. Right? The process of immunoprecipitation is a way to take toxins and viruses and other pathogens out of circulation so that macrophages, which is what monocytes become, can come by and eat them. Okay. Antibodies are also going to play a role in a process called opsonization, where they coat pathogens that are covered in proteins that come from the complement group and make it easier to eat those pathogens. Okay, and they're also going to promote the lysis of cells through a process called agglutination by using complement proteins as well. So they'll attach to antigens on the surface of an infected cell, okay, and then complement will attach to the antibody, and that'll blow a hole in that cell and kill it. Okay, so different methods, right? And then of course the monocytes, which become macrophages, just run around and eat invaders. So that kills them. Phagocytosis leads to the pathogen being inside the macrophage, and then lysosomes fuse with that and kill whatever is inside that vesicle. But it doesn't stop there. We take pieces of that particle and we display it on the surface of the macrophage as a warning to the lymphocytes that recognize those molecules that there's an invader in town and we need to take care of him. Okay? So that's the long and short of who these cells are and what they do. Okay? You can see here an example of some of the different cells. This is almost literally what they look like after they're stained. You can see the platelets and the monocytes and neutrophils and erythrocytes and so on. What I want you to notice is that the erythrocytes and the thrombocytes don't have any nuclei or organelles to speak of, and so they don't have that dark purple stain in the middle. That part of the right stain, it's the nuclear stain, doesn't light them up. And for the leukocytes, the white blood cells, what you'll notice is that you can see the nuclei in most cases, and the nuclei have specific shapes, and the shapes will help you identify the different leukocytes, along with clues that you get from whether or not you see the granules. Okay, so you can see an eosinophil bilobed nucleus, a neutrophil multilobed, okay, a monocyte kidney-shaped nucleus, and a lymphocyte nucleus that tends to be big and round. Okay, so that's part of the clues you can use to do what's called a complete blood count, where we take your blood and we smear it on a slide and we stain it, and then we look at it under a microscope that has a grid on it called a hemocytometer, and then we count the numbers of different types of cells in a particular area, and that gives us the percentage of cells of each type. And then by comparing that to known percentages, we can determine sometimes what's wrong with you. Okay, for instance, if you have fewer red blood cells than you should, you may have a condition called anemia. You have more red blood cells than you should, you have a condition called polycythemia. Okay, if you have too many eosinophils, okay, that could indicate a condition called eosinophilia, or it could indicate a parasitic infection or an allergic reaction. Okay, so that's what we call a differential, right? We look and we see how those percentages line up with normal percentages, and that helps us with the diagnosis. Okay, you can see here an actual smear, and the question is, let's identify what you see, right? So let's use the laser pointer, and what I see, let's use green. Actually, let's use blue. There we go. What do I see? Okay. Well, that's a lymphocyte. That's a basophil. That's an erythrocyte. That's a neutrophil. That's another neutrophil. That's an eosinophil. And that there is a monocyte. Okay. And the only formed element I don't really see here is a platelet. Okay. On this slide, we can see some different formed elements. The question before us is how many lymphocytes are visible in this field of view, and the answer is 1. There it is. But what are some of these other things visible? Well, there's a neutrophil. There's a neutrophil. That little speck there, that's a platelet. Those are erythrocytes. Okay. And of course, the stuff between the cells is plasma. Okay. Name 4 different types of formed elements seen here. Well, I see erythrocytes, a whole bunch of them, right? I could see platelets. You have to look kind of close to see them because they're tiny. I could see neutrophils. There's a neutrophil, there's a neutrophil, and I could see a lymphocyte down there. Okay. And of course I see plasma. Okay. So let's ID the cell and talk about what each of these types of cells do. Okay. So what's A? Well, A is a thrombocyte, also known as a platelet, and it's active in blood clotting. Okay. B is an eosinophil, and it is important in fighting parasites, eating antigen-antibody complexes, and in modulating the immune response through the release of antihistamines. A here is an erythrocyte. Erythrocytes transport oxygen, some CO2, and some protons. And the neutrophils, of course, engage in phagocytic attacks on bacteria. Okay. This is a basophil, and basophils initiate inflammation, okay, and also produce chemicals that are vasodilators and attract white blood cells to the inflamed area. Okay, they have a role very similar to cells that are called mast cells. Okay, mast cells are, you could think of them as almost fixed macrophages, right? Macrophages that are locked in place and can't move. Okay? These guys kind of drift around. B is another platelet, right? We already know what platelets do. And A is a monocyte. Monocytes become macrophages, and macrophages' job is to serve as what's called an antigen-presenting cell. So what they do is they phagocytose an invader. Lysosomes fuse with the phagosome and chop it into tiny bits, and those bits of invader get displayed on the surface of the macrophage as foreign antigen, and then those lymphocytes that recognize that particular foreign antigen are going to be stimulated to divide and make an army that will go out and kill anything that that antigen is attached to. Okay? So the kind of cells we would create that would attack based on recognizing these antigens would be B cells and T cells. Okay? This is a lymphocyte, and lymphocytes are agents of our adaptive immunity. Okay? This is the immunity that results from exposure to a foreign antigen, which generates not only cells designed to destroy anything that antigen is attached to, but also produces what are called memory cells, which can stay with you your whole life. And then the next time you encounter that same foreign antigen, you destroy anything it's connected to much more rapidly, and you don't get sick. That's why what doesn't kill us makes us stronger. Okay? List 3 types of granulocytes: eosinophil, basophil, neutrophil. The 2A granulocytes, lymphocytes and monocytes. What do red cells transport besides oxygen, CO2, and protons? Which of the formed elements differentiate into macrophages? That's monocytes. What type of white blood cell is the most numerous? Okay, and the mnemonic for that is never let monkeys eat bananas, where never stands for neutrophil, L stands for lymphocyte, E stands for eosinophil, and B stands for basophil. Okay, so never let monkeys, neutrophil, lymphocyte, monocyte, eat bananas, eosinophil, basophil, right? So the most numerous is going to be the neutrophil. Which of the white cells contain heparin and histamine? And that's the basophil, okay? Which of the formed elements are cytoplasmic fragments of megakaryocytes? And that's thrombocytes. Megakaryocyte just means cell with a big nucleus. Megakaryocytes are unusual because they undergo mitosis but not cytokinesis. So they have this giant nucleus, and they just have little cytoplasmic extensions that break off in blood sinusoids, and those little membrane fragments that break off, those are the platelets. So this is made in the red bone marrow. Which group of white cells include cells that produce antibodies? And that's the lymphocytes, right? Which are a group of leukocytes, white blood cells. And which white blood cells attack parasitic worms and eat antigen antibody complexes? And the answer is the eosinophil. What's a normal red count? 4 to 5 million cells per microliter. Rank the erythrocytes, leukocytes, and platelets from most to least numerous, and that would go erythrocytes, platelets, and leukocytes. Now, blood typing is something that we do in order to determine the compatibility of donor and recipient for the purposes of blood transfusion. Sometimes in the surgical procedure, you need extra blood to keep the patient alive. Okay, and if we don't give blood that is compatible, what's going to happen is that that patient is going to have a transfusion reaction in which his antibodies will attack the incoming blood and rupture those red blood cells, and the hemoglobin will be released, and that will block capillary beds, and that will cut off blood to downstream tissues, starve them of oxygen and nutrients, and they'll die, and the patient will die. Okay, so here's a little video on blood typing. Let's do this. No. And automatic. There we go. And stop. So, if we consider the ABO blood groups, what we're talking about here are a set of antigens that identify your red blood cells as belonging inside your body. So you could think of them as chemical ID tags that provide them clearance to do their job. In ABO blood typing, the antigens are groups of sugars on the surface of the red blood cells, and the antibodies that person will have will always be opposite what the antigens are. Otherwise, they would agglutinate their own blood, the red blood cells would rupture, and you would die. Okay? So, if you're type A blood, and we can use the, let's use the laser pointer here. Okay? If you're type A blood, you're going to have A antigen. That's the definition of being type A. Okay? The antibody that you have will be against anything but type A. So they'll have anti-B antibody, right? If you're type B, you'll have the B antigen and have the anti-A antibody. And if you're type AB, you'll have both the A and B antigen, and you won't have either antibody. Okay? People with type O blood have an antigen that has no complementary antibody in the human body. Okay? So the O group of sugars doesn't produce an antibody, right? So the antibodies that type O individuals have are going to be against A and B, all right? So when we think about a blood transfusion, what we have to match are the recipient's antibodies not cross-reacting with the donated incoming antigens, okay? So as an example, right, type O individual can only accept type O blood, right? Because if they were given type A, B, or AB blood, their antibodies would react against those antigens. The incoming blood would agglutinate and rupture. The hemoglobin would be released and they would die. Okay? An AB individual has no antibodies against the ABO antigens in his blood, and as a result, he can take any ABO blood group, right? And so ABs are called universal recipients, right? They can take A or B or AB or O. Okay? A type B individual can take either type B blood or type O. A type A individual can accept A or O. Okay? Type O can donate to any of the other groups. Okay? And the reason for that is that generally when we take donor blood, we resuspend the donor blood in saline and dilute out the donor antibodies so they don't become a factor. So the only consideration is the donor antigens and the recipient antibodies, right? So as somebody who's O negative, I'm O negative, I get calls to donate blood all the time, okay, because my blood can be used for any patient, okay? So you might think that people with type O blood are in a bad situation since they can only accept type O, but it turns out that type O blood is some of the most common blood in the country, so there's always plenty of it. So what we want to do in the next series of slides is ask some questions about the antigens and the antibodies and the reactions that we see here, okay? So if we look at this blood test card, all right, what we see is agglutination in both the A well and the B well. That's what that coffee ground appearance indicates, right? That's agglutination of the blood. So the way this test works is that whole blood from the patient is mixed with anti-sera that comes in a dropper bottle, okay? And if there is a agglutination reaction, that means that the antigen is present, okay? So this person is type AB. They have the A and the B antigen, and they don't have any ABO antibodies, right? So they are universal recipients, okay? This person doesn't have agglutination in either well. So this person is type O, okay? And that's because they don't have the A or the B antigen. They must be type O, right? So this is universal donor, okay? And then this person would be type A, right? So they would have the A antigen and the B antibody, okay? And they would be able to donate to somebody who's A or AB, and they could accept either type A or type O, okay? Here we see the same card, and the question is, how would the typing of a sample of type B blood differ in appearance from the typing on the card C here? And the way it would differ is that the agglutination would be in the B well here and not in the A well, okay? This is an example of agglutination, okay? Agglutination and coagulation are two different things. Agglutination is the clumping together of cells by antibodies followed by their rupture, okay? And that's because of interaction of the antibodies with something called complement, all right? Coagulation is a different phenomenon altogether. In coagulation, what happens is that chemicals are released from damaged vascular tissue that cause the attraction of platelets that release more chemicals that attract more platelets, and eventually that leads to a cascade reaction in the plasma that ends in the generation of a protein called fibrin, okay? And the fibrin acts like a meshwork that traps cellular debris and ruptured platelets, and that stops bleeding, okay? So that's a complement, that's a process known as hemostasis, okay? And it works, it's one of the few examples of positive feedback, all right? So the question here is, fluids like those shown in the dropper bottle were used to perform ABO blood typing. The vial on the left here, the one that says anti-A, contains what? And the answer is anti-A antibodies, okay? Fluids like the vials here were used to perform the blood typing exercise. The substances in these vials were used to detect what? And the answer is antigens on the red blood cells, okay? So that's how this operates, all right? What follows is a discussion about the function of blood, all right, and blood typing, okay? So stay tuned and listen to that, and I will see everybody in the next podcast. And I thank you for listening.
The circulatory system is responsible for more jobs than you know, and it does them by using only two hardworking divisions, the cardiovascular system and the lymphatic system. The first system we'll talk about is the cardiovascular system.
Section A, the cardiovascular system.
The cardiovascular system delivers oxygen and nutrients to cells and removes carbon dioxide and metabolic wastes from cells. The cardiovascular system is made up of the heart and blood vessels.
The heart acts like a pump, pumping blood through the blood vessels. Blood vessels are like the plumbing that carries the blood freely through the body.
Hi, I'm a blood collector. My name is Chuck. A lot of people try to describe serial killers as bloodthirsty. This is simply not true. Well, take me, for instance. I have no desire whatsoever to drink blood. I just collect it and store it in my refrigerator to be used at a later date as engine fuel.
When you take blood and you break it down, it's made up of two components. They are the plasma and the formed elements.
The difference between the plasma and formed elements is that the formed elements are cells and cell bits. Well, plasma is mostly water with dissolved stuff like proteins in it. So the formed elements are living blood cells, full of life and born to party. They're the erythrocytes, or red blood cells, and the leukocytes, or white blood cells. There's also the thrombocytes, or the platelets.
Erythrocytes carry oxygen, the leukocytes fight infection, and the platelets, which are actually cell fragments, these little buggers help with blood clotting. That means they help you stop bleeding when you're cut. All these formed elements are suspended in the plasma. Plasma is a non-cellular, straw-colored, sticky fluid that's 90% water.
Now you have to understand that blood does a lot of things. These things will be covered in greater detail if you take physiology. For now, just remember that blood is all about exchange. Exchange, exchange, exchange.
Blood's in charge of exchanging gases, hormones, nutrients, and waste between tissues. So blood is like a liquid cargo ship, carrying stuff to and taking stuff from tissues. Exciting, huh? Another thing that blood does is regulate body temperature. It does this by absorbing and distributing heat. But that's not all. The blood prevents infection with its white blood cells. And on top of all that, the blood also maintains proper fluid volume in the body.
Wow, the proper fluid volume. You don't know how important that is. Without blood, we'd be a dried up husk like the guy in my basement. And just think, we have so much blood, the normal amount of blood in the human body is 5.5 liters. That's 8% of our body weight. That's 5 times more than an average serving of cola.
Blood is responsible for transporting oxygen to the cells and removing carbon dioxide for excretion by the lungs. Blood carries nutrients, ions, water, hormones, and enzymes to all body cells and carries away the waste products. Blood regulates the pH and temperature of the body. Blood is composed of the liquid portion, plasma, and solids called formed elements. Plasma makes up 55% of the total blood volume, 91% which is water. Plasma proteins are albumin to maintain osmotic pressure and water balance, globulin antibodies to help the immune system, and fibrinogen, an essential component for blood clotting. The formed elements are erythrocytes or red blood cells, leukocytes or white blood cells, and thrombocytes or platelets. Red blood cells have no nucleus, therefore they do not divide and only live 120 days. Red blood cells contain hemoglobin that carries oxygen to all cells of the body and then carries away carbon dioxide. There are 5 types of white blood cells, 3 granular and 2 non-granular. Under the microscope, the granular types appear to have grains. They are neutrophils, eosinophils, basophils. Neutrophils phagocytize or eat up foreign substance and secrete lysosomes that destroy bacteria. Eosinophils produce antihistamines that combat irritants. Basophils are also involved in allergic reactions, secreting heparin, histamines, and serotonin. Monocytes also phagocytize bacteria and dead cells, and when they enter the tissue they are called macrophages. Lymphocytes produce antibodies and are crucial to the body's immune system. Bacteria are being destroyed by neutrophils, lymphocytes, and macrophages. Platelets initiate the chain reaction in clot formation.
The hematologic system is responsible for many functions, including the transport of oxygen and nutrients to, and the removal of carbon dioxide and waste products from the peripheral tissue. Disturbances to the hematologic system are common in the older person, and often the symptoms are vague and attributed to normal aging. Anemia or decreased numbers of red blood cells can be the result of nutritional problems, drug toxicity, chronic illness, and metabolic disorders. As anemia is a sign, not a diagnosis, further evaluation of the older person with anemia is always indicated. White blood cell production can be affected by disorders of the bone marrow, lymph nodes, spleen, and thymus. When lymphocyte formation is impaired, the older person will have problems with humoral and cell-mediated immunity and will be unable to mount an effective immune response. Platelet adhesiveness increases with age, and older persons are at risk for the development of deep vein thrombosis and peripheral edema, especially after suffering a traumatic injury, in the presence of atherosclerosis, or with changes within the blood clotting factors. Anticoagulation therapy is often prescribed for older people, and, with proper administration, monitoring, and support, can be a life-saving intervention. Many functions of the hematologic system remain constant in healthy older persons, including RBC lifespan, total blood volume, RBC volume, total lymphocyte and granulocyte counts, and platelet structure and function. Average hemoglobin and hematocrit values decrease slightly with age, but remain within normal limits.
Type 2 hypersensitivities involve interactions of antibodies and surface antigens of cells, followed by complement-assisted lysis of these cells. A typical example is mismatched blood transfusions. There are 4 different blood groups based on types of antigens on the surfaces of the red blood cells. People who are type A have A antigens, and those who are type B have B antigens on the surfaces of their red blood cells. Persons who are type AB have both A and B antigens, and those who are type O have neither A nor B antigens. The serum of people with type A blood contains antibodies against type B antigens, and the serum of people with type B blood contains antibodies against type A antigens. Type AB serum contains neither antibody, and O serum contains antibodies against both A and B antigens. If blood from a person who is type B is transfused into a person who is type A, antibodies present in the type A blood react with the surface antigens on the incoming red blood cells. This leads to complement fixation and lysis of these cells. If blood from a person who is type A is transfused into a person who is type B, antibodies present in the type B blood react with the surface antigens on the incoming red blood cells. This also leads to complement fixation and cell lysis. Persons who are type O lack A and B antigens on the surfaces of their red blood cells and are therefore universal donors. Persons with type AB blood lack antibodies against A or B antigens and are therefore universal recipients.